So this is the end of our first year of these research spotlights. We started in Spain, then we disappeared off to Australia, then we went to France, the UK, Denmark, and now we're going to Canada, where we're very lucky to have Heidi McBride join us from Montreal in Quebec province in France. So it's great to have Heidi here. I would just like to just highlight that you can put questions in the chat box and we will try and deal with those. I would also like to thank Supernus Pharmaceuticals, who supported our ability to do this. So thank you very much for allowing us to do this. And also to remind people that whilst this is our first year of doing these research spotlights, it's not the last year. So we will be beginning again next year. And I'll remind you at the end on January the 19th, when Ron Postuma from Montreal, so we're keeping it in the in Canada, will be joining us. So it's great to have Heidi with us today. Heidi is somebody who's done some fantastic work about trying to understand why cells go wrong in certain states related to conditions related to Parkinson's disease. Heidi is a cell biologist, but I thought it might be useful, Heidi, just to sort of start off and tell us a little bit about yourself. I mean, you're a cell biologist, so I'll ask that. What does that mean? Because for a lot of us, we understand a cell, we understand a biologist, but we don't know what the two together mean, and how you ended up being a cell biologist and interested in Parkinson's disease.
Right. Well, thanks, Roger, and thanks, Eli and the World Parkinson's Society for inviting me to this spotlight. It's a huge honor to be here. Yeah, I'm a cell biologist, which means basically I try to understand how the inside of a cell works, how it's organized, how things move around, how all of that stuff is regulated. And I've been working on a part of the cell called the mitochondria for many, many years. I did my PhD in mitochondrial sort of trafficking, let's say. And you know, this is the energy powerhouse of the cell, as people have learned in high school or elementary school now, probably. So when I started my research lab, the idea was the mitochondria are super dynamic and they do all these behaviors that nobody really understood why. So I began to understand or look into why do they fuse and how do they move and why do they divide? How, you know, all these kinds of just basic questions that a kid looking down a microscope, watching live movies of mitochondria, they're like little worms in a cell, would try to figure out how it works. And in the journey of learning those things, we sort of started running into, you know, trying to identify proteins that were regulating mitochondrial behaviors. And some of those proteins, it turns out, were mutated in patients with Parkinson's disease. So I kind of ran into it by mistake. And that's how I got into Parkinson's.
And so where did you start your career, Heidi? Have you always been in Montreal or have you been?
Well, I did my undergrad. I came here in 1986 to do my undergrad in biochemistry at McGill, and I did my PhD at McGill. And then I did a postdoc at the EMBL in Heidelberg, which is the European Molecular Biology Labs. Yeah. So this is a pretty famous kind of a very concentrated place of basic research. That's where I really got my cell biology legs. Before that, I was more of a biochemist. And then after that, I went straight to my own independent research lab at the University of Ottawa Heart Institute, where I was for 11 years before I was recruited back to McGill, where now I'm at the Montreal Neurological Institute, surrounded by neurologists in a complicated thing, working on the brain. So yeah.
So you've been there now for
10 years.
10 years, yeah. 10 years. Okay. And you sort of say that we'll come back a bit to discuss these mitochondria, what they do, because I think for a lot of us, we sort of have a picture of a cell with all these little things inside them, and they're there and they sit there and they do one particular function, and the mitochondria you have today will be the mitochondria you have forever, and that's that. It's not a very dynamic picture, whereas you're trying to tell us this is a bit more of a dynamic process, and that may lead to disease. But I think whilst your research has run into Parkinson's disease, I think Parkinson's disease has also, within your family, I think that's also been something that you've been, you know, very close to home. So I don't know if you'd be happy just telling us a little bit about how Parkinson's disease has affected you personally in terms of your family.
Well, I mean, it started with my grandpa McBride. So my father's father was a farmer. I mean, I grew up on a farm in Westmeath, Ontario. And so he had Parkinson's disease sort of back in the late 70s, I guess. And it didn't, you know, when you're a kid, you didn't pay much attention to it. He was an old man who was sick and you're a little kid. So, but then later my father and my uncle, so my dad's brother and my dad both got Parkinson's disease in their, let's say 60s, 70s. And they both presented very differently, which is quite interesting. So my dad had a lot of tremors, and he progressed much sort of quicker than my uncle, who had a lot of he was very rigid, sort of slower progressing. So that was an interesting kind of contrast within the same two brothers, how differently they presented. Which, as a scientist, you wonder, how is that? Obviously, it's not pure genetics. It's not like they have cystic fibrosis. There's some environmental or other factors going on. So yeah, I had already started working a little bit in my lab on Parkinson's disease when my dad was diagnosed. And so that was kind of an interesting coincidence, So
it didn't sort of drive your research. You didn't sort of say, well, now my dad's got it, I want to make sure my research heads in the direction.
It had happened before that. I mean, my own research interest really is to understand how the cell works. My dad always said, and I wrote this in the blog, you can't fix what you don't understand. And to understand things, you need to go right down to the beginning, to the nuts and bolts of it. So that's what I try to do. And mitochondria were a huge black box in terms of integration into the rest of the cell and how they really work and how they're signaled to, and like so many questions that had been kind of ignored because this organelle was so well known as a maker of ATP, and people.
So I mean, we all think of this, the mitochondria. You have lots of them in your cell. They basically, as you say, produce ATP. They produce the energy for the cell. And I suppose, coming back to where I was a few minutes ago, we'd sort of view them at they sort of static organelles. So these little organelles, little things inside cells that just produce energy. And as you get a bit older, they get a bit tired, like the rest of us, and eventually they stop, and we stop. And that's pretty much the story. And that, but you're trying to tell me that that's not the story. So that may be where it began when you started research. But these, I mean, I guess one of the questions would be, how do you keep mitochondria going, and what happens if your mitochondria start not going so well? What happens then?
Well, I mean, they're obviously turning over in lots of ways. There's a lot of mitochondrial biogenesis. So the new proteins are synthesized and put into the mitochondria all the time. And then there's lots of different ways in which proteins, when they get old, or lipids, when they get old in a mitochondria, are degraded to keep it clean, right? So a lot of us, and that's where that story hits into Parkinson's. There was initially, the work was saying that people with Parkinson's, or flies, model organisms with Parkinson's, would have problems with mitochondrial quality control. Okay. So the turnover wasn't happening as well. And so they were dysfunctional, and that was sort of considered.
So in a normal cell, you'd expect that the mitochondria, they just you're putting new things into them. The ones that aren't doing so well, you get rid of. So you just keep turning over and you basically have a healthy mitochondrial pool. And then in Parkinson's disease, that process wasn't happening normally. So you're ending up with more and more mitochondria that were unwell, if you like, and that was driving pathology or problems within the cell.
Well, people made the assumption it was driving pathologies, and they still do. And that's a big theory. Our own work, I would say that's part of the story, right? Okay. And I mean, the idea that entire organelles are turned over in a process we call mitophagy. This has been a big story. Richard Ewell won the Breakthrough Prize for this discovery, that two proteins that are sort of causal for Parkinson's disease, that they're playing roles in this process of mitophagy. Right. But there's other roles these proteins play as well. And it's been difficult to prove what you can see in a dish of tissue culture cancer cells is happening in a dopaminergic neuron in a patient. I mean, that's a big leap, so trying our own work we see evidence of roles in quality control, of course, like other people do as well, but also a lot of roles in immune signaling and other types of things as you start to look more specifically at pathways. So, I think the story is still open. I mean, I would argue.
So there's two things there which are very interesting. One is, I suppose we always, you sort of alluded to it there, that mitochondria are obviously in every cell. So if mitochondria go wrong, it's not just nerve cells that could go wrong, other cells could go wrong, which, if I've understood it correctly, immune cells and other things could be contributing. We've discussed that in previous sort of spotlight research, that Parkinson's and the role of the immune system. Yes. I suppose the other question I have is mitochondria don't sit in isolation in the cell. You say new proteins, new lipids come in and out. So who would be the major partners that mitochondria talk to in the cell? I mean, they obviously produce energy for everything, but do they have a close association with other little parts of the cell, other organelles?
Oh, yeah. I mean, that's a huge burgeoning field. And you have to understand that mitochondria don't just make ATP, right? They make hormones, they make lipids, they make iron sulfur clusters, they make, there's about 1,000 biochemical reactions in these creatures, right? Yeah. And they were bacteria a couple of billion years ago. So these were, evolved from alpha proteobacteria. So they were
they were bacteria that got inside a cell and just decided they like to live inside the cell.
Right, they got inside of the archaea.
Yeah.
And I have a lot of discussions with evolutionary theorists, and we think a lot about how you evolve the entire endomembrane system. So how did the cell that we know today, that has a nucleus and endoplasmic reticulum and Golgi apparatus and endosomes and peroxisomes and all of these things, how did that evolve, right?
Yeah.
From the initial acquisition of mitochondria into the archaea. So I know this is maybe getting a bit off topic, but so the point is, these are not just carriers or something that sits there and makes ATP. They've evolved. They do many, many, many things. They contribute many pieces of biochemistry, right? All of the intermediates of nucleotide synthesis, the purines and pyrimidines, are generated in mitochondria. Amino acids are built and degraded there. So these are really, really important organelles. And I feel like that was one of the problems as a cell biology entity. They weren't considered equally. And I used to get really mad. Like during my PhD, I was working on mitochondrial protein import, which is like we would, take mitochondria from a rat heart and add radioactive precursors to it and see how they would get put into how they would get sorted. But in the meantime, you have all the cell biologists, which is trying to figure out how proteins are secreted from the cell, and endocytosed, and Randy Sheckman and Jim Rothman and all these now Nobel laureates were understanding the flux of a cell and the interactions and GTPases and snares and all these molecules and machineries that govern cellular dynamics. Yeah. None of them looked at mitochondria. It was sitting over in the corner, right?
Because it was assumed that everyone knew what mitochondria did. They made energy, and that was that, and it just powered everything else that was going on.
There was just no interest, right? And in fact, when I did finish my PhD and wanted to do a postdoc, I wanted to look at mitochondria integration into the cell. How does the mitochondria has not been sitting there for 2 billion years and not paid attention to all this other stuff, right? There's no way that's true. There's no way that's true, right?
Yeah.
But I couldn't do a postdoc. There was no labs that worked on it. So I had to go to the cell biology field and learn about endocytosis and how that works.
Endocytosis is where things get taken up into cells. So things get engulfed and taken inside.
Right. Yeah. Because, to bend a cellular membrane is not a trivial thing. I mean, there's RAB GTPases, and to fuse them with another compartment, there's snares, and there's like all of this pathway. So I spent my PhD time deeply embedded in the core principles of cell biology, of membrane trafficking, of how membranes work. And then when I started my research lab, so middle of my postdoc time was about 1997, and Minx Fuller, who's a Drosophila geneticist, published a paper looking for mechanisms of male sterility in fruit flies. And which sounds insane.
A niche area.
Very niche. Well, actually, but she identified a large GTPase that she called fuzzy onion that was required for these sperm to be motile, because all the mitochondria in the sperm tail fuse into one.
Right.
If they don't fuse into one, the sperm can't swim, right? So then that was 97, then 98, Jody Nanari and Janet Shaw, and primarily I worked out that this is conserved in yeast, that in yeast, the mitochondria fuse, which was seen in like 1914 by Margaret Lewis, but nobody paid any attention to it, right? Okay. So when I started my lab in 2000, it wasn't even clear that mitochondria were this dynamic in mammalian cells. It had never really been shown. So I got a grant out of, I don't even know how. I had no preliminary data. I had a sequence of DNA that was similar to fuzzy onion from Margaret Fuller's paper, and I started my lab. So it was.
So this is really quite a new thing. I mean, I'm pretty ancient, and so I mean, mitochondria and POGSs has been around for a while. So I remember when I was training in the late 80s, there was this idea that mitochondria, which produce the energy, as we've said, and there are different complexes within it that do it. There was a particular one, complex one was down. So there was this link with mitochondria and POGSs, but it was the simple view, I guess, which is obviously where you've radically changed our view in this and the field, is that, the mitochondria had a problem with complex one. They couldn't produce energy. Cells were running out of energy. They couldn't keep going, so they died. And that's what happened in Parkinson's disease.
Yeah, but people always wave their hands, right, Roger? I mean, why would it be dopaminergic neurons that died? That's always the question. Why not all the other cells?
Yeah.
So
science. And why brain cells? Because, as we say, mitochondria everywhere. You sort of think, well, if they start going wrong, why doesn't the rest of you all just fall apart? And that clearly doesn't happen.
The heart should go wrong, and the heart needs lots of energy. But I think there's a lot of places where scientists tend to just allow a big black box in that they just walk over.
So when you started your lab or started really working on this 20 years ago, it was a pretty uphill struggle to persuade people that mitochondria were worth going back to look at, and that they had this sort of dynamic life, which you just didn't understand. And if you didn't understand it, you couldn't understand what could possibly go wrong in disease, which is not really what drives you in so much. It's you're trying to understand how the cell normally works. By understanding it normally works, you can then start to understand how it goes wrong in disease states.
Well, I would say it didn't take that long, because shortly after the mammalian versions of that fuzzy onion sperm tail thing in Fuller, shortly after they were discovered, they realized that those, especially mitofusin 2, they call it now, is mutated in a neurological disorder called Charcot-Marie-Tooth type 2A. Right. And then another protein that's required,
which is a problem with nerves in the arms and legs rather than anything in the head.
Right. That's right. And there was another core machinery of mitochondrial fusion called OPA1, which is mutated in autosomal dominant optical atrophy, which is sort of a type of blindness. And you would know more about that as a neurologist than me. But so there was a number of, like, as soon as we discovered started to discover the machinery that was regulating these dynamic behaviors, they were found to be, mutations in these proteins that were causing various types of disease. Okay. So then, in the mid 2000s, the fly people started to realize that mutations in PINK1 and Parkin had major mitochondrial phenotypes, and that turned it back. But your point about the original Parkinson's triggers of MPTP, so these are toxins that target mitochondrial complex 1. Everybody knew there was something about mitochondria in these diseases, but it's been, people are simplistic, and they think it's just energy. And the most important thing that I've tried to work on in my lab, I think, is understanding the signaling that regulates mitochondrial biochemistry, right? Mitochondria don't just make energy randomly. It depends on the signals, if the cell's dividing, if the cell's doing whatever, or, do you want to make amino acids or burn them? So are you in starvation, or are you growing? Like, there's all sorts of signals, and so it's much more complex than just the mitochondria sitting there making ATP and then they don't work.
Okay. So if the mitochondria are very responsive, and might I say, it would be lovely, do send questions in, be a nice responsive audience as well with the questions. What is it responding to? Is that signals that come from within the cell? So or is it signals that are coming from without? So you talked about endocytosis, where obviously the cell grabs something and brings it inside. It obviously can bind to things on the outside of cells' receptors and then trigger pathways. I would imagine it's a combination of the two. But what are the sort of major pathways you would say that sort of drive mitochondrial behavior?
Well, the original ones that we would look at would be things related to programmed cell death. That was the earliest side, when different pathways of apoptosis or ferroptosis now, or different types of.
So this would be where a cell gets a signal and says, "That's it, I'm out." And
well, initially discovered sort of in development, where the cells between your fingers would be killed off in a very clean, tidy way. It's part of a developmental process. So mitochondria hold the keys for cell growth and division. There's a lot of work now in stem cells that the mitochondrial metabolism will switch. That switch is really important for the cell fate change. Because basically, with these 1,000 biochemical reactions, if a cell gets a signal and needs to change its state, insulin and whatever the case may be, there's always an arm that goes straight to mitochondria and an arm that goes to the nucleus. You have to inform both of these beasts similarly, and often the mitochondria are informed first. So we see signaling platforms that we thought would just go through the cytoplasm, and people draw them in models with arrows. We thought, but actually they land on the mitochondrial surface. And the big one was antiviral signaling. So when you get into the immune system, there was a number of, James Chen at UT Southwestern was the first one of 4 people to discover this protein called MAVS, which sits in the mitochondrial membrane. It's essential for the cell to respond to viruses, right?
And so the mitochondria go first rather than the nucleus, because they're quicker responders. They can do things more quickly, whereas the nucleus obviously has to code things, and it might take longer for it to respond.
And I think evolutionarily speaking, the mitochondria, as old bacteria, were already signaling organelles. They were already receiving signals. They'd get infected by phage. They would do, as bacteria, they did all these things. Right. So, it's.
And I guess if you're a bacteria, your big thing is you want to survive. So mitochondria are pretty good at responding because you want to survive.
If you have an early stress in the cell, the mitochondria will hyperfuse. They all fuse into an interconnected reticulum. to protect themselves. And there's some really amazing behaviors that they do, right? So so when you say what signals do they respond to, I would say every signal. I mean, nothing happens in the cell without telling.
Okay. And obviously different cells respond in different ways to problems in Parkinson's disease. So I suppose, having heard this about what the mitochondria do and just what a sort of dynamic sort of center they are to processes within the cell. I mean, which you talked about Parkin, I think you talked about pink one. I mean, these are genes which are associated with quite rare forms of Parkinson's, often quite young onset. Yeah. But how do they relate to the type of work you're doing? So what sort of problems do they cause, those genetic abnormalities?
Well, there's two angles of that. So one of my the things I discovered in my lab was that mitochondria can generate vesicles. So they will, so they're of course like big bean-shaped structures and they're big tubes. They're like bacteria, but they also, like bacteria, can bud vesicles that carry selected cargo. Okay. And mostly that stuff are damaged proteins that go to the late endosome for degradation. So pink one and Parkin can promote that pathway. So as an early response to stress, and it will carry these things away. If the stress is chronic and very late, you'll get the whole organelle will go through this other process called mitophagy. Right. So one of the things is in my lab, trying to understand how these vesicles are formed, what regulates them, the signaling, how the cargoes are selected in those vesicles. But the other thing we ran into. So just on that, so the vesicles, so these come out of the mitochondria, yes, and they're containing, what do they contain from the mitochondria? They're things that it wants to get rid of, which will then be degraded through this dust cart, yes, through the lysosome. Yeah, because you don't want to turn over this whole beast. I mean, to take the whole thing down is a lot of energy, especially if you're in a nerve terminal. You're miles away from the cell body, right? You want to edit, you want to chew away and spit out the pieces. And the problem with mitochondria is because these biochemistry processes are large megadalton machines, right? Electron transport chain. The stuff in the mitochondria, pyruvate dehydrogenase, is like a megadalton. So when you want to, if it gets oxidized or damaged or it's too old, you can take it apart protein by protein, but these are many proteins in these machines. So we think we just spit them out in a vesicle, and the vesicle goes, and the rest of the mitochondria stays behind. So you're sort of editing it, right? Okay. And so it takes those out, and then they goes and gets rid of it. And so your question is, well, what governs? How does that process controlled, regulated? Yeah. And do those vesicles, these little sort of buddings out of the mitochondria, is it just rubbish, or do they use it for signaling? So do they sort of say, "Oh, well, actually, I've got something. I don't want to get rid of this. I want this to go somewhere else and do something useful." Well, I think it depends. So far we've been working on two different classes of vesicles. We call them mitochondrial derived vesicles, or MDVs. So one class goes to peroxisomes, and we think that's part of the biogenesis of the growing and building of new peroxisomes comes through. So what do peroxisomes do, Heidi? Are they? So peroxisomes are organelles in the cell. They do a lot of beta oxidation of very long chain fatty acids. They have a lot of catalase. So they neutralize reactive oxygen species, and they're thought many of the proteins that are in peroxisomes are also evolved from alpha proteobacteria. So they came with the mitochondrial lineage. Okay. We think the mitochondria kind of gave birth to this other organelle when it got into the archaea. But so peroxisomes are mainly, again, involved with sort of killing things off, getting rid of bad things for cells. Right. And I have to say, as much as mitochondria were understudied for a long time, peroxisomes are even worse. I mean, understanding what peroxisomes do in the nerve terminal there's hardly anyone working on that area, right? Mutations in peroxisomal proteins can cause very devastating human disease and Zellweger syndrome and stuff. So anyway, I work on peroxisomes too. But well, can I just ask you a question then? Because that's quite an interesting point. Because when we think of nerve cells, we tend to think of nerve cells. That's it. And you're implying that also what happens where the nerve ends up in a nerve terminal might not necessarily be the same as what's happening in the cell body, which is where the nerve actually originates. So obviously in Parkinson's, you have the cell body down in the substantia nigra and the terminals right up in the striatum. Yeah. So, I mean, I guess one of the questions which comes from this sort of is the pathology that you're particularly interested in, well, not pathology, but the process is, would it be different in a nerve terminal to a cell body? And is Parkinson's therefore likely to begin more in a nerve terminal than in a cell body? Or other? Well, I think, of course, I don't really particularly work on that. And some of the work that we've done we have a mouse model where we can get Parkinson-like symptoms, infection, which we can talk about if you want later. But we see a retraction of the terminals from the striatum. Cell bodies are intact, and the mice have profound Parkinson symptoms, fully reversed by L-DOPA. Right. So, and I think the dieback hypothesis is something that neurologists are talking about. So the idea is that it would start in the terminal. You know, that you have to maintain all of these things out there. I mean, Tom Schwartz at Harvard has done lots of math on how many mitochondria you need in one single dopaminergic neuron cell. Like there's thousands of them, and they have to be distributed. And it's an amazing amount of biogenesis to populate these arborized terminals through the brain with mitochondria and make sure they stay healthy and make sure they move and make sure that they're responding, you know.
And do you think that's where some of the selectivity lies in the cells that are susceptible to the disease process, because they have these huge, you know, numbers of connections and arborization, so terminals going everywhere, and to keep that, I mean, you know, coming back to the simplistic view, which is somewhat wrong, they obviously require a lot of energy to keep all those going, but actually it's a very complicated process. So trying to keep, you know, 15 different underground systems going is much harder than one.
Well, I mean, all neurons have that same kind of problem. I mean, dopaminergic neurons are more arborized, but I think to me, the you know, a couple of unique features of the dopaminergic neuron. One is, of course, iron, right? Yeah. They're heavy to make dopamine, which requires iron, and mitochondria are the handler and generator of iron sulfur clusters and heme and all these kinds of things. So there's a heavy reliance on iron cell biology that I think has been ignored. I think in the earlier days of Parkinson's research, there was a lot of focus on iron as something that's unique to those. And I personally feel that the iron cell biology is a disaster, and there's a lot to be done there. But the second thing I think is, you know, David Seltzer in New York at Columbia has said that catecholaminergic neurons, of which dopaminergic neurons are these, they uniquely can express high levels of MHC1, which is an immune molecule that normally is used to drive axon guidance, you know, and in development. But we think that that might be happening when you have a local inflammation in the brain. It might get overexpressed to reprogram and rewire the terminals when there's inflammation. And that would render them susceptible, because if you have high amounts of MHC1 and you happen to have a T cell that will recognize it, you can get cell death. So this is part of our theory. So there's a couple of different underlying features of dopaminergic neurons that we're kind of looking at among our team to figure out why that would be so unique there.
I mean, that's interesting, because obviously a couple of research spotlights ago, we were talking to Caroline Williams-Gray, who actually in the office next door to me, who's working on immune cells and is doing a trial of azathioprine. And so so there is this question about the extent to which the immune system may not cause Parkinson's disease, but certainly contributes to its progression. And I suppose that's what your work also would sort of suggest that. Another thing we stumbled into, you know, that whole project was basically my friend Michelle Desjardins was trying to figure out, and I don't want to get too much into this because it gets I mean, immunology is a nightmare. Okay. Yeah, it is. It's like, oh my God. Anyway, so he was trying to figure out how you have immunotolerance. So in the, you know, basically your own cell, your immune system is not allowed to recognize yourself. It can only recognize pathogens, right? And so the question is, how do you remove T cells that are recognizing self in the thymus? And so he was looking at the presentation of proteins from yourself to the immune system, so that it states you don't have autoimmune disease. Anyway, the point is that we discovered that mitochondrial vesicles played a role in mitochondrial antigen presentation. And the long story short, we did all this work, and the theory was that if you didn't have PINK1 or Parkin, you would have a lot more antigen presentation if you had inflammation or a fever or these kinds of things. But we were doing this all in a dish, you know, cheap cells that we work on. And so we decided that if this was true, we could take a mouse that's lacking PINK1, for example. Patients lacking PINK1 have early onset Parkinson's, right? But the mice, there's nothing wrong with them at all, right? So I was like, okay, mice are terrible models of disease and you shouldn't work on mice, blah, 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 blah. So we decided that we would take these mice and infect them with Citrobacter, which is a gut bacteria.
that gives you diarrhea, like people get it, you know? So we expanded our collaborative team to include Samantha Grunheid here. So we infected these mice with Citrobacter, and they cleared it all fine. Everything was fine. But we saw that they were presenting more mitochondrial antigen.
Right.
And then 8 to 12 weeks after the infection, they started to freeze.
Okay.
The mice were frozen. They wouldn't run down the pole, and they were like, you know, poke. Yeah. But you give them L-Dopa and they recovered.
Okay.
Right. So this means we turned an asymptomatic model with a relatively benign bacterial infection into a sort of transient Parkinson-like phenotype. So this was the first proof of concept that this can happen, but that brings in the gut, the peripheral immune system, the brain. So then you realize, oh my God, like which cell types are important here? You know, what are the contributions of all of that? So after those first couple of papers, we expanded our team, and we were really lucky to be given a big grant from the Aligning Science Across Parkinson's Society thing. And so now we've got card-carrying T-cell immunologists and neuroscientists and cell biologists like myself to try to piece this all together and understand what's happening in these models and translate it into human iPSC lines, blah, blah, blah. So there's, you know, but if I'm just trying to say that it's easy as a scientist to say, yeah, the mitochondria run out of energy and they die until the cells die. Yeah. It's like, isn't that nice? That sounds awesome, right? I've solved it. No. This is really complicated. And you need to have strong collaborations. Yeah.
Well, it's also less the sort of rather simplistic therapeutic approaches. You know, give creatine, give various things which are thought to be good to mitochondrial energy production, and then subsequently have not borne out in trials. So, you know, when you have these, you know, Parkin pink, and you have problems with, you know, mitochondrial behavior, we've talked a little bit about the fact that there is some selectivity with it. But I suppose the question would be, are the problems that we see in Parkinson's really unique to Parkinson's disease, or are these problems that you see in other sites? So there's always been this, you know, interest in things like LRRK2, which I know is slightly different gene, and that has associations with cancer, and there's a lot of interest in diabetes and Parkinson's at the moment. So I suppose one of the questions I would have for you, Heidi, is, you know, the extent to which your work on cell biology, whilst it speaks to Parkinson's through these genes, actually has implications beyond Parkinson's disease. How unique are these problems that we see in Parkinson's, or actually are they found in other disorders?
Well, yes, they're definitely found in other disorders, but I would also argue that Parkinson's disease is not one disease. I mean, listening to patients, and, you know, this whole argument about this umbrella term of Parkinson's, and like I said, even my uncles had two very different presentations, right? Yeah. So
if you were to study them, what do you think you would find? So if you were to get their cells and you were to look at their sort of mitochondrial behavior, would you anticipate that their mitochondrial behavior would be different or the same? Do you think they would have a lesion somewhere else within the cell?
Well, so one of the things, just to get back to your point on LRRK2, so other PD proteins, we've been looking at this as well, because part of the pathway, the cell biology pathway we've been uncovering in terms of this antigen present, whatever this stuff is we're doing. Yeah. It involves mitolysosome contacts and trafficking, and so a lot of if you mess up the lysosome, it's all part of the same pathway. So we think there can be commonalities here, but if you to look at, I mean, I think you had Per, I think on last time. So this idea of gut first versus brain first, this kind of stuff. I mean, my uncle Arthur could have been just brain first, slower lesions, more symmetrical and whatnot, and Dad could have had more of an environmental impact that just wiped stuff out. Like he had a valve blew in his heart, and he was on a respirator for a bit, and you know, that could have just wiped out a whole bunch of neurons that would have been just hanging on, and then after that he was much worse. So I think, you know, understanding the nitty-gritty cell biology of what I'm doing is obviously, I think, important, but there's other impacts, right, in terms of the patient experience and different types of traumas they have, and different types of the ways in which they're going to progress if they get big infections and fevers and things. These can rapidly accelerate disease progression. But understanding how mitochondrial response to infection and how they're controlling all of this stuff is important in the bigger picture, you know.
So I guess we can start to build a model where the cellular dynamics, what goes on in the cell in a disease state, is slightly abnormal, especially as you get a bit older, and that sort of it compensates to some extent. It can just keep going, and then an insult will come in somewhere.
Yeah.
And so it could be a genetic variant. So you could imagine your mitochondria aren't terrific, then your lysosome has a problem, and the two together now give you a real problem, and you start to get disease. Similarly, you could have mitochondria that are not so great, lysosomes that are fine, but then you get an infection, as you say, or you get exposed to some environmental toxin, and then that in combination itself now drives the disease.
Yeah, because most of those toxins and the infections are activating the immune system. They're activating inflammation, and inflammation is a very complicated cell biological process, right? It requires many parts, many moving parts. And I think, the problem is the fundamental mechanisms in cell biology are tricky. And, a lot of people who are like a neurologist or a cell biologist or an immunologist, they're looking at typing T cells. They're not looking inside the T cell so much. So people have different levels of granularity in how they study science, right?
Yeah.
So you can miss a lot of stuff and not understand it. So this is why it's a huge challenge to collaborate in these multidisciplinary teams, but it's also essential that you spend time to learn the language,
So I'm going to now move in a slightly different direction about how you study this. So somebody has written about, the complexity of the discussion. It is complex. Right. And how do we solve this? No. I think understanding that there's complexity is very important. But I guess one of the questions will be, we do epidemiology. So we look at populations and we come out with associations, and we sort of say, for example, certain types of tablets used for blood pressure, calcium blocking, antihypertensive drugs. People that on those tend to have slightly lower incidence of Parkinson's disease. Therefore, is blocking calcium an important part? Therefore, if we block calcium coming into cells and what effect it has, that would help people with Parkinson's disease. So we can sort of work back, if you like, from epidemiological data back to cells. Similarly, we can look at, your type of work, Heidi, and say, well, this is the cellular architecture we see. How can we look at that in epidemiology? So I can see these disciplines talking to each other, but I suppose one of the questions I was having is, how do we actually study this? So how do I know that your cell in the dish in your lab is relevant for the cells in the brains of people with Parkinson's disease? I mean, what sort of models we do? You've talked about fruit flies, you've talked about yeast, and we would sort of say, well, there's a big difference between yeast and the people I see in clinic, I would say, generally speaking.
Yeah.
Might not be with our political leaders, but certainly with the patients, I see it. So how do you study this, and how relevant do you think your models are for what we see in the clinic?
Well, I think our approach is really to have like a step ladder situation, right? So to make fundamental discoveries in cell biology, you can't use a very complicated cell type system because you spend forever growing them. So if you use patient-derived cells that you differentiate them into neurons in a dish, that takes many weeks, 2 months to get your cells. So you can't do a lot of hardcore stuff with that system. So we set up principles of we get the markers we want to look at, we get the cell biology outputs we want to look at, and then we put that into the next level of cell type, right? Into the neurons, into mouse models, patient-derived cells. We look for biomarkers and these kinds of systems. So I mean, it's So
you would sort of build it as that you would start off with something that's, people might not, but you have cell lines, cells that just grow indefinitely, and you sort of look at those, then you say, well, that's just a cell line, that's not even a nerve cell line, or if it is, it's a sort of not a brain
we do a lot of work in immune cells. Primary bone marrow derived macrophages and dendritic cells, we do a lot of work in that, taken from the mouse that have the lurk knock-in or VPS, different PD related mouse lines or patient lines that we get through the consortiums. So you feel
the confidence of what you're discovering and its relevance, I suppose, comes from, we've done it in this line, perhaps not very close to a human brain cell. We've now done it in another line, which is more like a human brain cell. We've now done it in some sort of invertebrate animal, perhaps, or in a mouse. They're all giving us the same readout. And that looks similar to what we see in the clinic. And can we get any information then from the clinic, which can inform us that this could be relevant?
It's beyond my team, clearly, right? I mean, like David Seltzer and, loads of people are looking at T cell integration into the brain, whether they're recognizing synuclein antigens or mitochondrial antigens or whatever they are. There's lots of evidence. We follow, of course, closely the evidence in people who are working right with patients, and whether our stories hold or not, But it's
so how does synuclein fit in with any of this? So is synuclein critical to your work, or would you say synuclein's sort of interesting, but actually doesn't speak directly to the type of things we're describing?
Well, I think in some of the newer work, of course, I wasn't really working on synuclein because I'm a mitochondrial person, but as we're moving into the more global pathway, of the lysosome and the endosome, and where you would have the breach to get cross presentation of antigens, these are all too many things to say in this thing. I think it will apply also, when you have inflammation and infection, it drives this antigen presentation. And if you're full of synuclein, it will present synuclein antigens, right? It depends. So some of the mutations, of course, we're working on pink Parkin mutations, but if you have other mutations, I think similar pathways can go forward, but it's just that the antigens will be different,
Okay.
But generally the process might be conserved, we think. I mean, I think that the evidence is indisputable that there's a massive role of the immune system in PD. Whether or not is a bona fide sort of autoimmune attack of the neurons at the end is unknown, but there's clearly.
So in your lab work, are you trying to, and I mean, I think you make a very important and good point about, I think when I started in research too many years ago now, you could sort of run your own lab and do your own thing, and you sort of just left your own devices. But nowadays it's team science, really. I mean, I mean, science has advanced so much, techniques are so advanced, and this idea, as you're putting forward, that to understand Parkinson's, you've got to have card-carrying cell biologists, someone who actually knows proper cell biology. They don't really care. They're sort of agnostic to disease in some way. They just want to know how the cell works. Yeah. And then you need people who have relevant expertise in disease. And then, as you say, you need people with relevant expertise in inflammation and how that interacts with disease states. So I suppose it's this bringing together of this expertise around a common problem, which I guess what ASAP, this new grant initiative has really done.
I also think you need patience. I mean, let's come back to this point. I mean, one of the problems with Parkinson's disease is that it's mostly studied by neurologists, no offense, right? And neuroscientists. But, if you talk to the patients, they have problems with constipation. They have, there's like microbiome issues, there's gut issues, there's things that bother the patients, They have sort of a dermatitis type of thing that goes on that the neurologist ignores because they don't care about that, right? But that can give you hints into autoimmune type mechanisms or things, that is beyond the brain. It's a whole body relationship. So I think listening to patients, and I've been on a number of various things where there's patient voices, and I think it's a really important reminder of things that are important to them. And also when I listen to them, and even watching my father and, being a caregiver to him through it, you notice all of these phenotypes. You're like, and I'm thinking about, so it's.
Well, I agree with you. I mean, I've often thought this, I think one of the big sea changes in the world of cancer has been that cancer is a systemic disease, which picks one particular site. And I would say Parkinson's is similar. I mean, this is a systemic disease. And obviously your work, every cell fundamentally does the same functions. It just has different emphasis on what it needs to do at certain different times. And these proteins are largely ubiquitously expressed, and so in fact, a
lot of them are more expressed in the immune system, right? I mean, LERC and Parkin are linked to leprosy and tuberculosis in human genetics, right? So there's links to these and inflammatory diseases on the side. So it's.
So do you try and model this in the lab? Obviously, you can get mice, and as you described earlier, you've got a mouse doesn't have any particular problems. You give it an infection, and suddenly it starts to develop Parkinsonian features. I mean, can you do this in a sort of cell bodies? Can you get nerve cells and then you add immune cells and see how the two talk to each other in a dish, or is that so artificial that it doesn't tell you anything useful?
Of course, everything's artificial, right? And unless you're a patient, unless you're, we can't cut the patients up right now, right? That's a terrible. We do. Of course, we do co-cultured work, and we're doing a lot of, we're moving a lot into. So there's two sides. Well, there's three sides that our team is doing. We've got gut things. So what's happening when you have the infection? Joanne Stratton and our team does single cell sequencing, and with Sam, who does the infections, we can figure out what's the difference in a pink knockout or a wild type mouse when you get the gut infection. In fact, if you look at the cells that are coming in, the immune cells that are moving around, the things that are happening in the gut, there's lots of differences, right? That Joanne is understanding. Same thing in the brain. So it's not just the neurons, it's the astrocytes and the microglia and all of these cells types, and who's causing the inflammation. Is there because the neurons get all their fuels and nutrients from the astrocytes, right? So,
Yeah.
and there's all sorts of interplay here between these cell types that we're trying to model. Yes. It takes longer.
But do you think there's a common problem with all of the cells? So I'm interested in this idea where you've looked at the behavior of mitochondria and their relationship to lysosomes and such like. And do you think that we could say, well, this is so complex, how are we ever going to solve it? You're saying there's a problem with neurons, there's a problem with astrocytes, the other cells of the brain, there's a problem with microglia, there's problems with the immune cell, there's a problem with the gut. How are we ever going to solve this?
No, we don't know.
Actually, do all of those cells have fundamentally the same problem, and together they add up and give you a disease?
Well, I don't know if there's a problem with the astrocytes, right? I mean, we have to, Mark Cooksen's done some really interesting cell biology work with astrocytes. And I think until we look, we won't know, but we can't make any assumptions, right? Okay. This is the problem. I think there's a lot of assumptions that get made all the time by scientists, and it drives me crazy because, there's no actual data for stuff, but because it sounds good and it fits into your narrative. It's like, it's, this idea of it's just quality control, I really don't think that's
true.
I
suppose I was guessing, what is it? If we sort of thought there was a sort of problem in some mitochondrial process and we could come up with some small molecule that could target it, it wouldn't just help the neurons, it could help all these other cell types. And by having a small effect, because I've always had this view that people look for these sort of, blockbusting drugs, but actually most people get to their 60s or 70s before they develop Parkinson's. They probably had it for 10 or 20 years before they present. Actually, you're quite good at dealing with disease.
Yes. Because
even genetic diseases, often people live for decades before they have any problems. So actually, you're quite good at getting rid of bad proteins and quality control in your cells. Absolutely. So actually, to make things better, you probably don't need to hit it too much in a different direction. So I suppose if you thought some of these processes were common to cell types and we could make a 10% improvement in all those cell types, actually, we'd have won.
Yeah, I agree. I think, but I don't think people should be discouraged by the fact that it's complex. I mean, we have AI moving forward. We've got so much omics. We can do things so fast, right? It's not going to be that tricky. The key is that people are willing to look under that rock, right? That they're willing to open their minds and try to understand it in a more holistic way. Yeah. And then we just move forward. I mean, scientists have done amazing things.
Yeah. Well, I'm a great fan of that. I always often say, I go to scientific meetings and people stand up and tell me all the problems in mitochondria and they sit down. Next person stands up and says the problem's all in the lysosome, they sit down. Next person stands up and says it's the unfolded protein response, and then they sit down. You think you could conceivably all be right, but it's always sold as though the only problem is this, rather than actually it's a problem with all of these. And if we can understand how they better relate to each other, we've got a much better way of actually solving this problem. And
it's always the problem of you only see what you're looking at.
Yeah.
Right. So you might be looking at the lysosome, but you're not looking at or vice versa, or how this is responding to inflammatory signaling. And you're not looking at NF-kappa B signaling if you're looking at like unfolded protein response. You know what I mean? That's like,
yeah. So how do you keep that, Heidi? How do you make sure you don't become the person who just always sees what they want to see and lives with the single idea that they've got?
You have zero confidence in anything. Right. I mean, you're never sure. And I think certainty, if a scientist seems certain, that's a problem. Yeah. I never trust a certain scientist. But I think you just do.
And do you think funding for research is helping that? So I think, for example, some of the recent initiatives of which we're both a little part of it has actually been very good at trying to break down those barriers, bring people together who work in slightly different areas. Because I could imagine 20 years ago, 30 years ago, you could have quite easily been going to your meetings on, mitochondria, and Parkinson's would be, thinking that's nothing to do with me. The immune system, nothing to do with me. But actually, these sort of, some ways, these chance interactions, these observations open up a new sort of horizon for your thinking and your science, and for other people who will come to you and say, like I just did, mitochondria just make energy and that's all they do. And you say, boy, you're so out of date. So this sort of capacity to cross-fertilize and work together, it seems to me this is a good thing we're doing, and trying to be challenging people on the things that they accept.
Oh, it is. Absolutely. But it's really hard. I mean, it's often like herding within our team, it can be like herding cats and people get in little arguments, and then, and then you're not rewarded for it. Like I've said this many times, like the way that we publish, right?
Yeah.
Your funding agencies and your promotions, they want the senior author in the last position, they reward you, and they'll reward you for the first position if you're a postdoc. But all the people in the middle don't get rewarded, right? Yeah. And so that makes it really hard to convince people to work with you on a story where they are going to really commit, when there really are only 2 positions on the page in the list of authors that will be rewarded, right? This is a massive, massive problem. And especially for junior investigators. I mean, like I've said, I mean, for me, I'm old and it doesn't matter. I mean, I'm fine, but I can be second, third, fourth from the end and it doesn't bother me. But for other people, it's a problem. So I think this is a major bottleneck in collaboration is the way that we set up reward systems and, metrics and the way science is. Yeah.
And for a lot of people here who are on this, webinar at the moment, who are not involved with science, I mean, people don't realize, I think, how hard it is. I mean, obviously I've sort of walked 2 paths, up a clinical path and a bit of sort of research, but you're absolutely right. I mean, you finish your PhD as you did, then you went and go and did a postdoc, and if that postdoc doesn't pan out and you don't get a 1 or 2 papers from it, you're now really struggling. And then when you set your own lab up, you say, well, I've got this great ambition as you did, I'm going to redefine what the function of mitochondria are. And after 3 years, they'll say, Heidi, what have you done? You say, well, I've disproved the following. And they'll say, well, yeah, but what have you shown? And you'll say, well, I haven't shown anything yet because I think it's all wrong. And then you're really struggling.
I mean, there's lots of ways to struggle. There's lots of ways in which we struggle. I mean, if you find something too new, you struggle.
Yeah.
Right? Like nobody believed that mitochondria shed vesicles for like 12, 15 years. They thought I was crazy. And I was only funded once for 5 years on MDVs, and that was it. So even if you do well and you've made a discovery, it's hard to get funding, and especially in a conservative place like Canada. But I think, I mean, at this point, I've done well. But there's a lot of challenges in collaborating, and I think it needs to be, the way that we think about science moving forward has to change.
Yeah. And how much do you think the patient voice can drive that? So, I mean, you made a very good point earlier on that we sort of, I think it's very easy in the sort of, I think it's very easy whether you're in the lab or you're in the clinic, that you sort of think, I know what Parkinson's disease is all about because I've seen it, I've studied it and such like. But obviously, we've just sort of said, we don't really understand it. We have very limited views on it. And often when you're talking to patients and patient communities, you get a whole new spectrum of problems and such like. I mean, how much do you think in this new world where we're trying to work together, the patient's voice should be more integrated into that?
I think that like I said before, there's a couple of reasons. First is actually to listen to the phenotypes, or I should say symptoms they have that matter to them, right? That neurologists will overlook because it's not a neurology problem that the patient is telling.
You can't do anything about it. That's always the problem.
But it's a huge clue. But it's a huge clue, right? To the etiology of the disease. If the patient's telling you, I have this symptom, you're like, oh, I don't care about that symptom. They do, right? The second thing patients bring to the table is common sense. Right? Like I've seen Ben Stetcher, who's like one of the premier sort of patient advocates. I've seen him at meetings, and he gets up and he will just tell Virginia Lee, I don't believe this. What are you talking about? Synuclein fibril spreading. And he's, it's fantastic what he does, right? And because he challenges us to face our dogma. Right? What is the evidence in the dopaminergic neuron that you have a problem with mitochondrial quality control in vivo, right? Pink and Park can regulate that. There's a lot of problems with these theories. And so to be held to account by patients who are brilliant people who have had long careers, who are, they may not be scientists, but they know how to solve a problem. And, like my dad was a mechanic, right? And a farmer, and he solved problems. That's what farmers do every day, right? So I think to be held to account is really important, especially to push us off our dogmatic hills.
I always say that. I think the most difficult questions I ever get are from the patient community and from children. Because the children will often ask you a question to which you think, well, that's a very simple question. And actually, now you ask it, I haven't actually the faintest idea what the answer is, because I've just taken that as assumed, but I don't actually know.
Exactly.
So it's,
right. But I mean, I also think part of the failure in Parkinson's therapies, I mean, dopamine was discovered in 61, or I don't know what year it was, but, and the failures in Alzheimer's, I might just see thing on the chat about Alzheimer's.
Yeah.
Again, the reason why it's been so focused on the A beta tangles and removing that is because it's run by neuroscientists.
Yeah.
Right. So these things have to be faced head on with an open mind and with humility.
Yeah.
Right. This idea of certainty in science is, everybody wants it with the vaccines, for example. I mean, scientists by nature are not certain because it's complicated. You have to account for a lot of things.
And I think, one of the messages I'll take away from our chat this evening or this afternoon is that, the more you study something, in some ways, the less you understand it. It just gets more complicated. So you sort of think, well, there must be a simple solution to this to understand it. And there is a logic to it. And there's an increase in knowledge as we study this, as you've clearly demonstrated with your wonderful work. But every time you discover something, it opens up another series of questions as to what that all means and where it's going. So I suppose
that's called progress.
Yeah, it is. But it's wonderful to think that actually every time I come in, because I remember when I started coming back to us 25 years ago, I think I'll have run out of questions in a year. I've only got a few questions and then you realize that actually as they start to become a little clearer or a little less relevant, you've got a whole new series of problems that you hadn't thought about when you start. So it's a very dynamic process in much the same way.
And that's like when postdocs leave my lab to start their labs, I'm like, take all of this with you, right? Because I got 7 million other questions, right? And they should take whatever they can to start their own lab and give them as much as. So these people who like hold back stuff, and the trainee who's starting their own lab after you has to kind of find their niche. It's like, no, you built a niche in my lab. You just take all of that, right? I think that's an important thing because there's so many questions and there's no.
Well, you're very generous. I mean, we're sort of coming to the end now, so I should just wrap up. But I mean, it's been terrific having this chat to you, Heidi. I mean, I think one of obviously the great skills you have is your wonderful enthusiasm for things, your ability to explain it, and your generosity. Because telling your postdoc to take it all and go off and do something does involve a generous spirit. There are unfortunately some scientists who will say, well, that's my area. You can go and do whatever you like, but just don't touch the mitochondria, don't touch the lysosome. You can't have the immune system. But you sort of think, I mean, it's obviously a very selfish bit, but your generosity of ideas is a wonderful thing to behold. The fact that you're prepared to just keep challenging yourself every day and think that what I published last year might be wrong because I've got the wrong idea on it. The data's fine, but my interpretation of it may now need to be changed. Yes. I mean, very inspiring for me, very inspiring, I'm sure, for everyone who's been listening here. And the fact that you've had all of your personal journeys with Parkinson's disease, you've seen it firsthand, and you stuck to what you wanted to do, which is the other thing I admire about you is you stuck to what you believed was interesting. And I always say this to young researchers, just stick with the topic. At some point you'll be flavor of the month. Everyone will want you to talk. Everyone thinks it's there. 10 years later they'll think, you're still working on that? I thought that was a dead field, so
well, everything old is new again, right, all the time. But I think thank you very much for all your kind words. It's been great to be here today, and I wish I could see all these things. Farmers do rule, Grant. See that in the chat. I think I don't know. There's a lot to learn, and I think being humble and being excited about the future of the technologies we have. Honestly, as soon as we open our mind and allow all the work to go forward in a concerted way, not leaving out things just because of the narrative.
I think keeping open-minded, working together, and realizing it's the big community, which of course is what the WPC is all about. It's everybody has a role to play. Patients with Parkinson's disease, scientists, clinicians, everybody. So thank you, Heidi, and thank everybody for joining us this afternoon. As I say, the next one of these will be on January the 19th in the new year, and it will be Ron Postuma, and he's going to be talking about prodromal Parkinson's disease and REM sleep behavioral disorder and what's the link between the two. So thank you again for listening, and thank you so much, Heidi, for such a terrific hour's conversation.
Thank you, everyone, for joining. Thanks.
Bye-bye now.
Bye.